Welcome to this video on ATP synthase, oxidative phosphorylation or synthesis of ATP. This topic is central to understanding energy metabolism and builds directly on what you have learned in the electron transport chain. You have known that electron transport chain creates an electrochemical gradient across the inner mitochondrial membrane. ATP synthase, also known as protein complex 5 of ETC or electron transport chain, utilizes this gradient to synthesize ATP. In this video, we will examine the detailed structure of this fascinating molecular machine called ATP synthase and its binding change mechanism underlying ATP synthesis. Let us explore the detailed structure of ATP synthase. ATP synthase is embedded into the inner mitochondrial membrane, which separates intermembrane space from matrix. ATP synthase is made up of two key components, FO and F1. FO is an oligomycin sensitive and is rotating structure that forms proton channel. FO is generally made up of 8 to 15 C subunits, which form a ring-like structure known as C ring. However, in mammalian mitochondria, C ring is made up of 8 C subunits as shown in the present picture. In addition, there is an A subunit present close to FO segment which perform important function to direct the, the flow of proton through C ring. In one complete rotation of 360 degrees, number of proton flowing through will depend on the number of C subunits in the ring. Now let us discuss the structure of F1. F1 is present in the matrix. It is stationary and catalytic. It is hexameric, made up of 3 alpha subunits and 3 beta subunits. Alpha subunits are purely non-catalytic, but they help in the binding of substrate, whereas beta subunits are catalytic and they perform the function of the synthesis and release of ATP into the matrix. F1 and FO segments are connected with a structure known as central stalk or gamma handle, which is made up of two subunits, gamma and epsilon. The central stalk or gamma handle and FO segment, they are rotating structures. F1 is also connected with another stalk known as peripheral stalk, which is made up of one A subunit, two B subunit, and one delta subunit. Peripheral stalk is stator that prevents the rotation of F1 head. However, rotation of CS or central stalk through F1 head causes conformational changes in the beta subunits through loose, tight and open, allowing thereby binding of the substrate, synthesis and release of ATP respectively. One complete rotation causes synthesis of 3 ATP molecules, which is equal to the flow of 8 proton or 8 hydrogen ions in mammals through C ring. Therefore, you should be able to calculate the number of protons required to synthesize one ATP molecule. By now you know the detailed structure of ATP synthase, so we should explore the mechanism by which this enzyme synthesizes ATP. The mechanism is known as the binding change mechanism or binding change hypothesis, which was proposed by Paul Bayer, who received Nobel Prize in 1997. According to this model, the three beta subunits of F1 head exist in three different conformations irrespective of the flow of proton or hydrogen ion. These are open or O conformation has low affinity for ATP and therefore allows the release of ATP into the medium. L is loose conformation, which allows the binding of ADP and PI substrates. It will not allow the synthesis of ATP. And the T or tense or tight conformation, which supports synthesis of ATP. However, it will not allow release of ATP because it has highest affinity for ATP. In the absence of the flow of proton, The beta subunits do not cycle through these conformations, and ATP synthesis and release are inhibited. In fact, ATP is trapped in the T conformation. However, when proton flows through the FO segment, it rotates the gamma handle, which forces each beta subunit to change its conformation, allowing therefore the binding of ADP and PI in the L state, synthesis of ATP in the T state, and release of ATP in the O state. A full 360-degree rotation of gamma handle produces 3 ATP molecules, or you can say 1 ATP molecule per beta subunit. This means that every 120-degree rotation generates 1 ATP. In the next slide, we will explore these conformational changes in one complete rotation of 360 degrees. Let us examine how gamma handle rotation through F1 hexameric head causes conformational changes in its beta subunits, leading to the synthesis and release of ATP. F1 hexameric head is made up of 3 beta and 3 alpha subunits. Beta subunits are catalytic and exist in one of 3 conformations, namely O or open, L or loose, T or tense. Open allows the release of ATP because it has the least affinity for ATP. L allows the binding of the substrate, it does not produce ATP. T or tense is for the synthesis of ATP, it does not release ATP into the medium. The release is through open conformation. At any given time, even in the absence of a flow of proton through FO segment, beta subunits remain bound with ATP and ADP and PI but do not synthesize or release, because synthesis and release is tied with the conformational changes caused by gamma handle rotation. Before I explain the conformational changes, let me explain to you that in one complete rotation of 360 degrees of gamma handle, 3 ATP molecules are produced. Therefore, 1 ATP is produced every 120 degrees of rotation of gamma handle, which is powered by proton flow into the C rings. So let us see what happens in the first 120 degrees rotation. Gamma handle will align in this position here, and there will be change in the conformations given here. OLT, T will change to O, T will change to O, and O has least affinity, therefore ATP will be released from here. O become L, allowing binding. This O will become L, allowing binding of substrate here, and this L will become T, leading to the synthesis of ATP. So in the first round of 120 degrees rotation, there will be synthesis of the first ATP molecule. In the second round of 120 degrees rotation, this will align, the gamma handle will align in this position, and again there will be changes in this conformations, in the conformation shown here. T will become O, thereby liberating ATP into the medium. O become L, allowing binding of ADP and PI, and L will become T, making ATP. So in the second round of 120 degrees rotation, second molecule of ATP has been produced. In the last or third round of a 120 degree rotation, the gamma handle will align in this position, and there will be change in the conformation again. T will change to O, thereby releasing ATP. The third ATP molecule is released here. O will become This O will become L, leading to the binding of ADP and PI, and the L will become T, synthesizing ATP for the next cycle. So this is called as binding change hypothesis or binding change mechanism of ATP synthesis, which is caused simply due to conformational changes. There is no input of any extra energy into the system. Thank you very much for watching this video. I request you to like it, share it and subscribe it, and please watch it in conjunction with the TCA cycle and electron transport chain. Until next time, we take care. Bye bye.